I'm not going to talk about my goat farm days, but if you need to know about farming, I thought it would actually focus more on the stage that you all are at, postdoc and young investigator stages. So I'll tell you about what my lab does, but you'll see the journey has been kind of rickety and torturous, and I hope it will kind of help you if you get through into some of these strange territories. Maybe this can help. So what my lab works on is a process that I discovered, which is how epithelial cells die, called extrusion, and then we'll kind of see how that becomes important when that becomes misregulated. So before I tell you how epithelial cells die, I think it's always important to tell you what they actually do when they're alive, because that's very important about how they go about dying. And so this is just an epithelial layer in cell culture, and you can see that these cells work collectively together as sort of a city to provide, the main point is to provide a protective barrier. And that's obvious when you look at your skin. I mean, you're looking at it all the time. You can tell that's a barrier, but it's lots and lots of layers. It's important to note that all of your organs are coated with a similar barrier layer like this. It's only 1 or 2 cell layers. And that is really essential for the form and function of the organs. And it's also important to think about it in terms of the innate immune system. So this is really the first line of defense for any types of bacteria, pathogens, things like that could try to enter the body. And so it's safe to say that if you experience any inflammation, any infections, there's probably been a breach of this barrier happening. And that will become important, but we're thinking this could be a new thing to think about in terms of inflammation. So really, when you're looking at this, it just sort of looks like bricks in a wall. This doesn't seem like that's very interesting to study. Like why is that hard to get through that barrier? But it becomes a lot more interesting when you think about the fact of the matter is that epithelial cells are turning over at some of the fastest rates in the body by both cell division and cell death. So just for instance, the place where they divide the most rapidly is in your gut. So all the cells lining your gut can all turn over within only 3 to 4 days. So this is like hundreds of billions of cells dying every day in your body. And so this is like hard to connect that idea that there's so much cell death and so much cell division at the same time when you're supposed to be having a barrier. So one of the first things that I looked at, and this is in my postdoc work, is how do you maintain this barrier in the face of so much cell death? How is that actually working? And then secondly, kind of the back of my brain, but then we got into this more, is oops, sorry. How do they naturally die anyhow at this very high rate? And more importantly, how are those cells that are dying connected to the cells that are dividing? Because with this very high rate, you can imagine that if you just sort of set a rate for cell death and set a rate for cell division, things could go awry pretty quickly. And so we sort of imagine there's probably some sort of link. And I'll tell you that's mechanical. But it's also kind of important because if you lose that, if you lose that link, and the number of cells that are dying exceeds those that are dividing, you could easily have barrier function problems. And that is, in fact, what we're finding, that maybe some of these immune diseases are really a result of that. But on the other side is that if you have too many cells dividing and not enough dying, then you could get cancers. And nowhere is this more important than in epithelia, because this is where 90% of all cancers form. And what I was sort of surprised with when I was thinking about, so one thing I didn't really know, I was reviewing a chapter for molecular biology of the cell, and they were saying, well, cancers are really when cells divide that don't normally divide, and they just divide out of control. And I thought, well, that's weird. I don't know, because I never thought about it like that. Really, it's the cells that are dividing the fastest that are ones that are most prone to getting cancers. And so we really thought that this whole idea then is really important, how you maintain those constant numbers, because this is really when cancers occur, and we'll show you how that happens later. So first, how did these cells maintain a barrier when the cells are dying? And to look at this is what I did when I was a postdoc. So this is the your acronyms, PDF. This is when I was a PDF. And what I did is I didn't have time to wait to see how cells naturally died. I just put them in a strata linker, and just caused extensive cell death. And these are MBCK cells in culture. And you can see that this cell is going to die. And as it does so, it pops up out of the layer. And while that's happening, the surrounding cells are moving into the space where that dying cell once was, so that no gaps ever form. So this is the process of extrusion that I found. And we found that even when as many as half the cells are dying and extruding, they can still maintain a really tough barrier, really tight barrier through this process. So how this happens then is that very early on, this cell that's going to die sends signals to its live neighbors that form an actin and myosin contractile ring, and they will contract around and below and really heft that cell out there. And there's contraction going on in the cell that's dying as well. So then, this is the answer. How is this barrier maintained? Well, the cells get extruded. So before really much death can be inflicted, they shove these cells out of the layer, and while that's happening, they repair the space where the dying cell once was. So that seems like a very nice, tight story, and it's believable. It got published, hooray. But the way I found this really had nothing to do with looking for this, answering this question. Really, how I found it was that it was just a distraction when I was looking for something else. And that is that when I went to do my postdoc, I went to work on wound healing. And so I went to this lab where they were studying how wounds heal in embryonic tissue and epithelia again, by this actin-myosin purse string. And so I went making all these wounds in different types of embryonic tissues. And I just couldn't focus on it because I kept seeing all these little wounds that I hadn't made. And I just kept thinking, what is that? Why is it? I'm sure I didn't make those. And after a while, I realized that this was probably just these cells that were dying in the background. And whatever wound healing was happening was channeling into the same kind of signaling pathway. So these were really just these apoptotic cells that were extruding. And so I went on to follow that. What I will tell you that is a bit more of the backstory on this, though, is it wasn't that easy because I was working for this guy who really wanted me to study wound healing. And I got a grant to study it, and he got a grant. And he really wanted me to obey and just study this wound healing stuff. And first off, he wasn't very happy by the fact that I was making wounds and making movies, and they weren't closing in the way that he wanted to. So already he wasn't happy about that. And I was feeling a little confused about that because I thought, I'm not directing the cells. It's not like I'm a director and the cells are actors. This is just what's happening. And then when I went and presented this in group meeting on this epithelial extrusion, he didn't respond very well. Actually, he said, "This is shite." And I was like, "What's that?" And because we don't use that term in America. So I went, "Oh yeah, that's what I thought it meant." And so I was kind of taken aback, and I wasn't quite sure what to do about this whole thing because it's really hard to do a postdoc when you don't have the support of your PI. And so, I was really, there was a lot of soul searching going on, and I had to, think about the other problems I was having was I didn't have a place to do cell culture, so that was a problem. I didn't have, the microscope I was using was in a well-lit office, my fluorescence microscope, so that was also a problem. I had to keep telling people, could you turn off the lights, and they'd get really mad at me. And so, and they were also taking a lot of my money away from my fellowship. So I went and talked to Martin Raff, because I didn't really know anything about apoptosis, and he said, "Oh, this is really great stuff. You should really follow it." And I just, and I was feeling so despondent about things. But eventually what I realized is that I had my own funding, so that was really important, that I could just decide, you know what, I'm just going to trust my own eyes instead, and I'm just going to follow this thing. And I had the support, and I moved over to a different lab, and really did somewhat of an independent postdoc. So I was working with Louise Cromer, who in her lab space, and with Martin Raff before he retired. And so I kind of was just a free agent. It was really actually the best time of my life. But it was a hard time, and emotionally I realized that this was I could really recognize this, I guess, when I went and took a picture for an ID, and I had this picture that looked very much like this. And I just was like, oh, why can't I make a proper smile? What's going on with me? And I knew there was a lot of problems. We just moved to London. I had a young baby. I had another kid that was in a daycare, and they were in 2 different parts of the city. And my oldest was having temper tantrums, and my husband was working in the lab where I was, so he was not all that supportive of my situation either. And so I was really in this sort of distraught mood, and I also had this sneaking suspicion that my whole career was over with. And then I just started thinking, this is it, I'm not gonna be able to make it, and I think I'll just be a school teacher. And what I realized then was that I was actually just taking myself way too seriously, and I had like all this pressure on myself to get this thing done, and I needed to, and I'm working, and I just thought, once I decided I could just be a teacher, it kind of all went away, and I started having fun again, and then all the results started to come in again. So I tell this to a lot of students because they get really worried, like, what lab am I going to choose? It's really, and I just think, it just doesn't matter that much. If it doesn't work out, switch things. It can work. It's stressful at the time, but you can do it. And I did it, and it was good. And I think it also is just a key thing to just have some levity and to just work hard, take yourself. And people have said it before, but I think when you start taking yourself way too seriously, it just it's like those experiments you do Sunday night for group meeting on Monday morning. They don't work. They just don't work. So that was one of the things I learned. And then from here, I moved to Utah, University of Utah. So I had a bunch of different job offers, and I think that some of the discussions I've been hearing about kind of feed into this. So I was born in Utah. I'm a Utahn. I'm half Mormon, half Jewish. I'm Mormish. So it's kind of a weird place to be. And I love the mountains. I'd really miss the mountains, but I felt like kind of going back to a place where I had family. That was one thing, but also a place where I felt like I could be more politically involved and kind of take charge and affect my environment a little bit. And also, it was a bit more of a challenge, this place, because it was the only cancer research place I was looking at, and I knew nothing about cancer. And I think I mentioned that, but that has been really good for me because I learned a lot. And so I think that was one of those things that it was like, I could go to a lot of other developmental biology departments. I actually really wanted to go to, I had a job offer in Cambridge that I really wanted to go to, but I just thought having the kids there and all that. So I think one of the things that's I've really liked being at the University of Utah because I feel like there's been a lot of good collaborations there. People really help each other out, and there's a good community effort. But those are kind of things I think when you're choosing where you're going to go, I'm sure I could have gone to a lot of places and they would have worked out well. But I think one of the things that I was excited about here was people there was a bit more space to be a little eccentric. You didn't feel like you needed to be in a place that was just really cutthroat and you had to have your cell papers every year. And what I was working on was totally new, so I needed some space to do that. So while I came there, I went and I started my lab, and we found the signaling pathway that controlled this. But in the back of my head was this question that I still had is like, how do these cells naturally die on their own? And this is surprisingly a topic that people haven't really looked at that much. People really study cell death in response to, I'm going to kill you, how do you die? It's like a homicide, here you go, I'm going to shoot you, now you die. And it was surprising people hadn't really looked at this sort of, natural causes sort of cell death. And really, when you go to meetings, like whole meetings will be on cell death, and whole meetings will be on mitosis or cell cycle or something, but these two weren't really tied together, and we thought this is so important. We need to really think about this together. So what I did then was just follow, this came really from intuition. It was just something that I noticed in the back of my head was that the places, I'm not sure you can see this, the places where we would see cells extruding most often were usually very crowded. And it wasn't something I had actually formalized in my head. I know it looks obvious here, but it was just like I kind of knew where to look. And when I started studying it in a scientific way, where I actually measured it, what we found was that the places where they were extruding were always 1.6-fold more crowded than the places where they weren't. And now we've had a recent paper that shows that places where cells are dividing are also 1.6-fold more stretched than the places where they're not dividing. So there's this nice golden ratio of death and division by crowding and stretch. But this was just an observation, so we needed a way to test that. And so what I did was make a series of different types of stretching devices that we could use in reverse. So here's just this, this is just a stretch plate you can buy commercially, and we have this manifold to stretch them on. So we'll grow them in a stretch state and then release them from stretch. And what you get is you can see these cells are crowded now. Whoops. There we go. By 30 minutes, you can see that they're crowded, but by 6 hours, they go back to densities that were seen in the control. So as if these cells had a certain, a sense of personal space in the way, well, each type of epithelium would have a different type of personal space. This might be more like India, and this is more like Utah. and so what happens in Utah, is that if people get too close to you, kind of go, ooh. And so we needed a way actually to test this, and we started looking at different model systems to test this whole hypothesis in. And what we turned to eventually was humans. And this is a human that's my daughter's friend, Blake. And these are my daughters here. They're dressed as nuns for Halloween. And so this is them at a really nice density that they prefer. This is just them hanging out. But when they go to this party later, or even worse, if they go to our public concerts in the summertime, they're free, then you can see when Blake gets really crowded, she just gets extruded from the monolayer. So that's exactly what happens, is that we saw that when the cells reach a density that they prefer, if more cells divide, which they're constantly doing, this creates crowding stress. And we now know the whole pathway that drives this. But that will return the cells to this optimal density right here. So, so then the next thing we wondered was, how is this crowding actually causing cells to undergo apoptosis? And when we looked at this, so we just, again, looked at lots of different types of epithelia. And what we found when we stained for cells that were dying with this caspase-3 apoptotic cell marker, this is in the adult human colon, but we see the same thing everywhere, is that when cells are dying, when they're undergoing apoptosis, they're extruding, but most of the time we saw cells were extruding when they were actually alive. And we've gone on to show that these cells are not just caspase negative, but they really are alive. So really what's happening then is that this isn't a homicide. This is not a suicide. Cells aren't undergoing cell death. They're not just like lying down and dying like we once thought. They're really getting killed by their neighboring cells. The neighboring cells are shoving them out, and then they go on to die through this process called anoecus, which is death due to loss of survival signaling, or death due to homelessness, which is because they no longer have the contact with the matrix and the neighboring cells that gives that survival signaling. So this is really then that cells are getting shoved out because they're too crowded, and that kind of makes that link pretty well. So, so the final part that I want to talk about is what happens when this extrusion is disrupted. And if what we're saying is really true, if we really are right, and we've really found the thing that drives epithelial cells to die, then you might expect that there would be a lot of problems if this gets misregulated. In a work I won't show you, we're finding that if there's too much extrusion, as in, if there's too much crowding, too much extrusion, as in asthma, then you lose that whole barrier, actually. And we think that's what causes the downstream inflammation. But here, what we did was look and disrupt extrusion. And what we found, actually I haven't said this, but a lot of our studies we're doing in zebrafish because we can see everything that's going on. So every time we've been looking at things in culture, we feel like we have to make sure that that's really happening in bodies. So what we've turned to routinely, not all the time, but routinely is zebrafish because they have this nice epithelium here that you can see. It's just a bilayer of epithelium. And when we were looking at a mutant, a naturally occurring mutant that disrupts this extrusion, what we found is that, yes, the cells, when they were targeted to die, couldn't extrude. But if you just looked at them over time, what they did is they formed these huge masses at sites where they should have extruded. And so this was true, that if we disrupt this extrusion, now these cells can't go anywhere, and they have this prolonged survival signaling. They just build up these masses. And this was to kind of prove the point that I thought, oh yeah, you really do need these cells to extrude, to die. But what my postdoc who found this, Yapeng Gu, who found the extrusion signaling pathway in the first place, he went on to look in different cases of cancer. And he found that a really aggressive class of cancers, pancreatic cancer and some types of lung and colon cancer, also disrupt this extrusion process. And this makes them very resistant to chemotherapy as well. So based on that, we also started to wonder, well, what about extrusion as a way of maybe activating cells to leave the epithelium and metastasize? And what we got interested in is this idea that if the cells are extruding, but they're not dying initially, what happens if you block that step of anoecus down the road? Because we knew that metastatic cells often have upregulated survival signaling that blocks that whole anoecus part. And so this is work then that really comes from a fantastic graduate student I had. She was a technician in my lab first and then went on to do her graduate studies in my lab, Gloria Sladham. And the thing that's great about her is she was a little bit naughty too. And she also had this disobedience, which is what really led to this whole wing of my lab doing this work. And what she found is that this aberrant extrusion could cause, disrupting the extrusion signaling could cause invasion. And what that is that she, so there's this idea. So we wanted to put in some kind of oncogenic mutation to see if we could block the subsequent cell death. And she wanted to put in KRas, and I told her, don't do this, it's going to be boring, because we had colleagues who'd done something similar and found not really that interesting results in my view. But she did it anyway. So she just did all this work, and she said, I'm seeing something weird. And that weird thing is that when these cells, she would put it just in, if even at this cell alone, she could see that they were cell autonomously basally extruding instead of apically extruding. And they were also surviving. So now if you think about it, if these cells are extruding out apically, even if they don't die, they kind of are lost. They're just sort of in this, in the lumen of your gut or something like this. But what she found is that instead, if they're extruding basally, they could maybe start to invade the rest of the organ that the epithelia is encasing. And so we needed a way to look at this in vivo. And what she turned to then is zebrafish. And so really she developed all these tools in zebrafish to start looking at this problem. She hadn't worked on zebrafish before. And what she found was that if you put in this oncogenic KRas mutation just on the outer surface of this epithelium, you could see that there's actually far fewer of these cells than if you just tag with a membrane tether, GFP, and express that. But they also form these masses, as we saw previously when you disrupt extrusion. Now, if you focus in on these cells, you see they have a very different behavior. So these control cells are just sitting on the surface. They don't really do much. But the cells that are expressing the oncogenic KRas mutation will extrude at a much higher rate. And when they do so, they go basally again, most of the time. So in this case, there was nothing but just this mutation, this one oncogenic mutation. And you can see that many of these cells that will extrude will go on to die. But she put them with a commonly collaborating mutation in P53, which is usually common with all of these types of KRas-driven tumors. And what you can see now is that there's lots of internalized cells here. And so if you look at this rotating movie, you can see that this is the sort of bottom layer here, but in P63 are these dotted cells, the stem cell layer. And the KRas cells start on the outer surface, but then they end up on the inside here. And so you can see that we're seeing a lot of internalized cells, but the beauty of zebrafish is that you can actually follow what's going on. And so in these movies you can see now, you can see lots of these cells basally extruding. So this one goes, this one goes. These ones I didn't orchestrate again. I didn't direct these cells going through, but they just go 1, 2, 3 in a row. And then if you follow them later, you can see that they're migrating throughout this zebrafish. So this is the whole cell surface. And it was also an interesting finding too that there's these masses that are occurring at some sites in the zebrafish, but the places where the cells were extruding really had nothing to do with that. So it really went against a lot of the paradigms that these tumors will form, and if they get big enough, then they'll start to they'll kind of walk away and start to metastasize. In this case, we're just seeing them quickly ducking out and going through. And so what we really think we found is then this new mechanism for tumor invasion, and we're just trying to put this paper together right now. So really, in summary, what we found with this extrusion is that when cells are targeted to die, if you get a bacterial infection or you get chemotherapy, we know that they'll maintain this barrier by causing the cells to extrude. But more importantly, most cells, this is the way most cells in the epithelium die anyhow, is that they are extruded while they're still alive, and then they go on to die as a result of the extrusion. Now, if you disrupt this, you'll cause masses to form at sites where they should have extruded, but you also get some cells that are starting to extrude basally, and they can just start to invade. And we think this is a new mechanism for how tumor cells can invade. And so now the benefit of this is having this whole system in zebrafish is now we can screen using inhibitors. And we have better ideas now because we know how they should have died. So we have some candidates, but we can also use this whole system to screen for inhibitors and maybe finally target these invaded cells, which has been always the problem with treating pancreatic cancer, because people, when they get their diagnosis, they already have metastatic cancer. So in summary, I just wanted to go over some of the hallmarks of, that was sort of the science story, but the other things I really wanted to highlight was pay attention to your distractions. So yes, I have ADD. My daughter has ADD. It can be a problem, but it can also be a good thing if you find things that you keep getting distracted with, as long as you can focus on them long enough to make a story out of it. Intuition, I think this is something that is not stated very often in science, but I think it's important. And I also, it reminds me of a conversation I heard, an interview from David Byrne and from the Talking Heads with PJ Harvey. And he said, do you know what you're talking about when you're in most of your songs? Do you have any idea, what your songs are about? And they both said, no, I have no idea what they're about. Not until 5 years later, usually. And I think that that's it. There's been a lot of these things that I've noticed in the back of my head, and I kind of made a mental note of it, but I didn't really know what it was that I was actually talking about. So those things can be quite informative, and it's hard to know when to follow your intuition or not. But disobedience, I think I've hit on this a few times. I think for us to really carve the paths that we want to go into, it's important to be able to say no. It's important to be able to say no to committees and things like this, but it's also important to remember where it is you want to go. And I think it kind of is the final thing I want to talk about in terms of when I've started my whole career, that I keep saying to people that work for me is keep your eyes on the prize. So, you get pulled off into a lot of different directions, and you get told, you have to do this, you have to do this for tenure. You have to keep remembering, okay, that's what I'm supposed to do, but what do I really want to do? And so I think that's important. And I guess I'll talk about a couple ways that that's come into play since I've had my lab. And one of them was that, in fact, it wasn't that easy to get a job based on a new finding. And I think that's something we kind of need to talk about a little bit, that it's not that easy to come up with something new and then be able to get grants and jobs on this. And so actually what I got my job for, I think, was really a completely different finding that was also a distraction, but it was about how myosin was required for spindle assembly. And I was really excited about this project as well, but I have to say that I think it got more attention because it was in, I'm almost done, because it was in a field that was already, well studied, right? And so I don't know that I had to get that other paper, but I think that in terms of sometimes getting funding, that may be true. And that was definitely true. Like I was told, when I started at the University of Utah, I was told you need to write a grant that makes it look like you work on epithelial cell adhesion. And I thought, but I don't do that. And so I wrote this grant, and it didn't even get scored. It got a terrible, it got panned. And I talked to the granting officer. He said, you've had this new discovery. Is that really what you want to do? I said, oh no, I have no intention of doing any of those experiments. He said, yeah, I could tell. It didn't look like, so you really, what I was lucky is that I got this had just started this innovator award. so I could really take off and do this work. Had I not had that, I'm not quite sure what would have happened, because a lot of times the buy-in price for a new R01, I'm not sure how it works in the Indian funding culture, but you need a few papers on something before you're going to get funding on it, and that wasn't the case here. So I could just kind of start and do this whole new thing, and it was great because I could actually just talk about what I wanted to do. So I think you know, but for me it was kind of like a non-starter. I felt like I had to do these things that I was really interested in, or I wouldn't want to do the science at all. And during this whole time, people were talking about what you need to do to get tenure, and I was like, I don't even know that I want tenure. Stop talking to me about that. I need to figure out how I'm going to make this happen. So in the end, I'll just thank all the people in my lab and the fund, you know, both past and present, and my funding, which I wouldn't be able to do any of this without. So thank you.